Welcome to our webinar series for professionals.
Well, let me do the introduction, and nice to see those, as I normally say, nice to see those who are live and show their faces on the Zoom camera, and to those who doesn't want to do it, just nice that you're there anyway and participate in the chat as much as you can and want to. So welcome everyone. And I'm Anne-Catherine, co-founder of Nordic Laboratories and DNA Life. And as I normally say, we are dedicated to change healthcare through functional medicine and personalized medicine. And you're all practitioners, so you should all have your VMS account. So check into that. All the products that we talk about now are there. Reach out if you need help. So I'm just gonna do a little bit of a news alert here, because on Monday, we're going to change our payment gateway. We are going to change to Stripe, and hopefully they will help us make all the payments hassle-free. We have been challenged on that, but that's because all credit cards are getting these double security, what have you. But Stripe should be what we need. So hopefully patients and you as practitioners will benefit from that. And then, as some of you already know, because of Brexit, we had to move our warehouse, start a warehouse in Holland as well. So we had to register a company with everything that follows. And when you don't speak Dutch, that's not very easy, I can say, but we have succeeded. And we are proud to say that everything in Holland is working very nicely. But the final piece in that puzzle is that we also have to change the currency, because right now all European customers see prices in Great British pounds. But hopefully on Monday, this will change as well. So all European customers will be seeing prices in euro for supplement purchasing. So that we are very excited, but of course, bear with us, but it should be quite a smooth process anyway. So today, Helen Gauci is the star of the show. And Helen is my dear colleague. We have been colleagues for 10 years now, I think. She's a registered dietitian, just like I am. And she has a Bachelor of Science in Dietetics. And at DNA Life, she's the one who develops the test, and she does research into all the SNPs that we offer and develops education material, and just really do a lot of work in order for DNA Life to produce new tests and be more scientifically based. And she has set this very strict criteria for allowing a SNP in a test. So there has to be a certain amount of publications, and the SNP has to be actionable, meaning you need to be able to do something in order to change the outcome of the destiny of the SNP. She's doing a very good job there. And some of you have probably already met her if you've been on some of the DNA Life education. And in just a little note on that, remember in your account under education, there is a link to further education. So if you want to look at some of the DNA tests that we have, Grow Baby, DNA Resilience, DNA Smile, from that matter, and then we're doing these the base or foundational courses as well, and we keep sending out information about those, so you can sign up for that if you're interested in that. But please chat in the chat box, and if I can answer your questions, I'll do that. If I think it's a question that goes for discussion at the end, I'll pull it up there. So, but for now, I will just pass on the stage to Helen. Here you go.
Thanks so much, Anne-Catherine, for inviting me back to this great session. And nice to see so many practitioners joining us. This evening, we really are going to, as the title of the of the presentation suggests, get back to the basics of DNA health. Today we're going to be discussing what DNA health is, why it's useful, considering its clinical utility, and how to use it in practice. So really getting back to the basics of understanding the DNA health test as almost a foundational test, and especially if you are interested in looking at the nutrigenetics and the genetic side of functional medicine and personalized healthcare. I always recommend DNA health as this foundation test to always use alongside some of your other tests as well. So this first slide and image really just highlighting all of the very complex pathways that we are looking at in DNA health, genetic variants in these genes that encode proteins and enzymes, possibly hormones, that are then associated or linked to these specific pathways. And if you look at all of these pathways, you should recognize quite quickly that they are all linked, so we cannot look at anything in complete isolation, but also that all of these pathways, when you are looking at the research around epidemiology and the cause of disease or how to improve health, all of these pathways play a very important role. And so this is where we are looking at in the DNA health test, and that is why I always suggest it as this foundation to health test, because we're looking at variations that play very important roles in each of these pathways, from your lipid metabolism to inflammation and oxidative stress. And you can almost start seeing how if you were to pull these pathways to get, to actually almost zone in and then look at pathway on pathway, how they so integrally linked. We look at methylation and detoxification, bone health, your insulin sensitivity, vitamin requirements that actually play significant roles in all of these major pathways, as well as specific food response genetic variations that give insights into possible intolerances and also possible risks if you are taking in those foods and you carry variations on those genes. So that's really, in a nutshell, what you're going to get from the DNA health test. And why this test is so useful. This is where we look at who's going to benefit from the test. Who, if you see someone walking into your practice, you do their history and you just get a good foundation of what they're all about and why they're there. These are some of the reasons why you would recommend this DNA health test. So we're looking at improving longevity, and I think we should put good longevity in there as well. So not just long life, but really good life. And where we're looking at that is by understanding genetic variations on these genes governing these pathways, we're able to identify where there might be possible weaknesses in these pathways that might lead to an individual feeling just slightly less than optimal, slightly less than healthy, looking at risks for chronic diseases that often end up happening much later on in life. And there's just so much research long, years back going into the recommended dietary allowances for nutrients to decrease or eliminate diseases of deficiency. But going forward, we're actually looking at not just diseases of deficiency, but what is actually optimal for an individual to live a good life. And that optimal for an individual is so varied based on who you are as that individual. And a part of that comes in from your genetics, from what you've been exposed to in utero, and from your lifespan, what environmental interactions you have, and what other possible health issues you might have. So looking at these targeted nutrition and lifestyle interventions based on understanding the biochemistry because of the genetic variants that we carry. Another one is where someone comes to you with certain functional tests or blood results, and there are discrepancies or there are issues in what you're seeing. There are little red flags that are coming up around possible dyslipidemia, higher homocysteine than we would like. We see that there might be some chronic inflammation or altered nutrient levels in serum or plasma, whatever it may be. And so all of these little red flags from what is happening here and now should give you an indication of obviously what their environmental exposure is, but also let's dig a little bit deeper to understand what is happening upstream that might be influencing these biochemistry results in the here and now. And so really useful to understand at that biochemical level, again, what is going on. For any individual who comes to you with a chronic disease from the diseases that might be, or the disorders that might be a little bit more difficult to actually pinpoint a diagnosis, and just by being able to regulate those pathways, you can just get much better health outcomes, from the chronic fatigue syndromes to fibromyalgia, certain gut issues, all where you might see DNA health giving really good insights into these areas. For individuals with mental health concerns, we do have our DNA Mind test that is a very nice addition, or can be used alone for looking at mood and cognition, but you would also get fantastic insights from DNA health. And so a very nice one to do, just giving those immediate insights into inflammation, your lipid metabolism, methylation, what is going on in those pathways. And then for anyone who is worried about developing a chronic disease that one of their family members might have developed, from your cardiovascular diseases to cancers, diabetes and metabolic syndrome, osteoporosis, periodontitis, all of these actual chronic diseases of lifestyle have a very strong genetic component together with what we're seeing in the environment. And so by understanding what is going on, again, at that biochemical level, because of genetic variation, you are able to regulate where there might be weaknesses to improve outcomes and really target your interventions. So that's kind of the why one would use the test. And now getting into more detailed here, actually understanding how to use the test, what you would get from the test if you were to open up this report, and then how would you use it in practice? What benefit would you get from it? So I'm going to go through a few little steps here. First, with the interpretation of the report, first in understanding these impact factors that we use. I think that's, if you understand that, there's a lot that just follows on quite easily from there. And then going into some examples with quite a basic case study, just to give you the clinical utility of it, and how one would interpret the results in practice. So starting off, you would get your report, and hopefully you would be familiar with what the report might look like. And if not, you can always get quite easy access to that. So that should be something that is quite easy for you to get. I will have some slides coming up soon of detailed information in the report that just gives you an idea of what you would actually get and what you would see. But one of the things that we speak about is understanding impact factors in the report, and that's what I'm going to go into now. So when you get your DNA health report, you would see there's a big table, and you'll see it on the next page or so, but there's a big table full of all the genes that we are looking at, and I'll speak about that SNP selection criteria again that and Catherine did already mention. But what you'll see is the gene name being given. So in the column, you'll see, the first column, you'll see the gene name, and this is quite a familiar one, hopefully to many of you. So that's why I've just chosen it as something that's quite familiar. Then the genetic variations that we can see on MTHFR. So at position 677 or at position 1298 of the gene. And when I speak about position on the gene, if you think of your gene as it's your DNA strand as this twisted ladder, and if you start at the bottom of the MTHFR ladder and you start climbing all the way up until you get to the step 677, that's where we're looking. And on that step, do you see a C or do you see a T? Okay, so that's simply put, but that's what we would be considering as at that position, what variation could occur. And so at 677, there's a C or a T, and at 1298, it's quite a long one, you could have an A or a C. And so for this individual, they've come up at 677 with a TT genotype, and then you see that we have assigned this high impact for that. And at the other one, the AA, because of what we've seen in the research, we've assigned a no impact to that. So your impact factors, no impact, low, moderate, high, or beneficial. And so when we're looking at a report, we want to understand sort of why it was assigned that impact factor, and what does that mean to us clinically? So we always want to focus on the moderate and high impact factors, really, because that is where we can then get an understanding that there might be something going on in that pathway at that genetic level that isn't as good as we would want. And so that's where the targeted interventions come in. But otherwise, and then when it's a low impact or a no impact, it means it's actually closer to the norm. It's closer to that genotype functioning the way the general population enzyme functions, and it is not related to any specific altered phenotype or altered disorder or altered health outcome. So that's where we say, okay, well, those genotypes, so on AA, that's not leading to any issues, but the TT, we do know because of that high impact, then we can look in the report that is going to lead to a change in how that enzyme might function or how the gene might express. A little bit of just interest on TT specifically, that high impact is related to a 70% reduction in that enzyme function. So that's where we would say, okay, because of that significant reduction, that's why it's getting that high impact. And if you were to go and look at the literature, what would you find? There would be thousands of articles on MTHFR 677C to T telling you all about this T variant and environmental interplay. So that assignation of impact factors, just to be clear, is a qualitative assessment. So that the assignation is actually qualitative, but it is based on the literature, which is obviously quantitative in nature. So the strength of the impact factor will be determined by the importance of that gene in a pathway, the strength or the amount of research for a specific SNP related to that SNP, and the significance of the SNP's effect on a given phenotype, taking into account environmental interventions. And I'm going to go through each of these in a step-by-step process with an example. So again, another big one that hopefully you should be familiar with, not just because of genetics, but it's just spoken about so much, is looking at, in the area of lipid metabolism, ApoE, encoding your apolipoprotein E. And then another little one, LPL, is your lipoprotein lipase, which if you're familiar with any kind of lipids or lipid metabolism, that comes up to a very important enzyme as well. So for this pathway, we see that for the ApoE, when we get the risk variant, we're assigning a high impact, whereas with the LPL, in this case, the variant actually gives us a beneficial impact, meaning that that enzyme is going to kind of counteract some of the negative effects that you might see in that pathway. To highlight, and I'm not going to go into these, but just importantly, just to give you the kind of comparison here, we've got a variance in the ApoE1 variant, one of the risk variants. So you've got an E4, and the E3 is kind of considered the normal, but you're still getting a high impact factor. Whereas with ApoC3 or CETP, you can see that they're carrying two of the risk variants, but they're only getting a moderate impact factor. And so why does that occur? So why have we given ApoE such a high impact factor compared to the other ones? So going through the step-by-step process, just remembering, I just want to take a step back, importance of the gene in the pathway, strength of the amount of research related to the gene, and the significance of the variants in that for that given phenotype. So starting with number one and looking at ApoE, the importance of that gene in the pathway. So this is actually taken, this image is taken from a paper from years back, but it's just a really nice way to show how ApoE, when it's functioning well, has very, very important effects on mechanisms downstream related to health. So when your apolipoprotein E, it's very important in the transport and metabolism of cholesterol, triglycerides, and other lipids and constituents within other organs, including the brain. But when we're looking at the functioning of this protein, when it's functioning well, it actually communicates with your antioxidant enzymes downstream. So it has very good communication skills and communicates to these enzymes, these antioxidant enzymes, to work well when you've got a nice functioning ApoE. Also, when your ApoE is functioning really nicely, it's related to or been associated with inhibition of your T cell activation and proliferation. It promotes cholesterol efflux. It inhibits your smooth muscle cell migration and proliferation, and it suppresses your vascular cell adhesion molecules. So all of these things that are actually, if you think of suppressing, inhibiting, all of the things that are kind of pro-inflammatory and atherosclerotic causing, it's actually controlling it. So when ApoE is functioning nicely, it's controlling a lot of these processes. And so if you think of just this one gene, the importance of that gene in that pathway is significant. It has major roles to play. It's kind of a boss or a star gene in the pathway. It has many important roles to play. And so the first reason we've included this gene and the reason we give it so much emphasis is how important that gene is in the pathway. If we then compare to lipoprotein lipase, we can't disregard the importance of lipoprotein lipase in the transport and the breakdown of your different of your chylomicrons and then eventual transport to the liver. And then also just looking at lipoprotein lipase also in the endogenous pathway and in cholesterol efflux as well, where lipoprotein lipase plays a role. So it's very important, as I mentioned, so in breaking down your fats, and then these fats get transported to the liver. And then from there, again, from the liver, it's involved in these endogenous pathways, breaking down and then transporting to eventually transporting to different tissues that need these different fats. So again, extremely important in cholesterol metabolism, but possibly this is where the change comes in. So both very important in the pathway, and that's why we've included both of them. So when we're looking at genes to include, they need to play a significant role in a pathway. We need to see if we're looking at this gene, if we intervene there, is it going to have a downstream effect? Is it going to actually change the outcomes for the individual? So that's, both of them have that. The second thing, though, is then when we look at the strength or the amount of the research for that specific variant or that single nucleotide polymorphism. So when we're looking at ApoE, this isoform that gets formed from the variants on ApoE, the E2, the E3, or the E4, if you go back, I mean, this is, I've got 2013 here, but you can go back to 2000, the early 2000s, where ApoE the literature started just accumulating. And the amount of literature, if you just go into one of the medical databases, so this was just from PubMed, there were over 4,000 genotype phenotype articles, and over, when we're specifically looking at our now specific areas, genotype environment phenotype articles, we're looking at over 150 articles there. So all in nice peer-reviewed journals, and we can then understand the strength of the research for this specific genetic variation is very strong. So when we're thinking about, okay, well, if we're thinking about assigning an impact factor, it's going to be quite a strong impact factor. And then just giving as LPL, which gives the beneficial impact, but slightly kind of less on the importance scale compared to ApoE. We do see some very nice articles coming up with lipoprotein lipase as well. But in terms of the genotype phenotype articles, they're around 100. and still quite a lot with regards to nutrigenetic studies because they are quite complicated to perform. Still enough considering our criteria, but if you consider it against ApoE, ApoE definitely comes out as the star gene. So just going into quite a bit of detail around the impact factors, but really just giving you the importance of understanding that significance of the genotype variation and how we assign these impact factors. And it's not just a thumbsuck. There is a logic behind it when we are assigning these impact factors, and just really trying to emphasize the science behind it, that we are really trying to make sure that when you are looking at a DNA health report, you are getting information that is valid, that is really science-based. And so just really highlighting conclusions from the research. We can see from the ApoE variants, it's been related to very important phenotypic outcomes. And when we're looking at these genotype, environment, phenotype studies, these nutrigenetic studies, there have even been some, more recently, some very nice intervention studies done on how we can intervene on the specific gene. And because of the genotype, by putting in targeted diet, lifestyle, nutraceutical recommendations, we may actually see positive outcomes coming out related to that E4 variant. So just again, highlighting that you need not just the gene with a variant, just some random variant, you need to see the functional change in it. You need to see that it's related to a specific health outcome or specific biochemical marker that you would want to change. And then you would want to see that there is something you can do about it, and the evidence is there to do something about it. So just really emphasizing those important parts. Sorry, then I've just also got related to LPL, and again, just some information on that. I'm not going to go through all of that in detail, but it is there in the slides for you to look at. But just understanding why this was now given a beneficial impact, it's associated with actually improved HDL cholesterol levels, and very nicely responsive to lower dietary saturated fats and moderation in carbohydrate intake to get the further benefit from that variant. So again, genotype, environment, phenotype relationship, and targeted information that you can action for your client. And so this is just before we then get into the actual report, just understanding why we choose the variants or the genetic or the SNPs that we do. And Catherine actually alluded to all of this. I'm not going to spend a huge amount of time on this part, but we need to see that the evidence is clear regarding a gene environment effect. It can't just be a gene phenotype effect. There needs to be something going on with the environment, because that's how we empower our patients with changing what their environment is. The variants must be relevant. There must be the gene variant needs to exert some kind of direct influence over a specific biochemical process that creates known symptoms or disease. And the environment should be able to be modified to gain a positive effect. And so we can look at gene expression, and so that's where the nutrigenomics comes in, or we can look at how we can maybe put in a little bit more of a nutrient because of the nutrigenetic interaction, showing that the requirements for a nutrient might be higher. So then with all of this, these should be measurable. So the impact of your clinical interventions to modify the expression of genes or change the eventual outcomes should be measurable through biochemistry or other biomarkers. We need to see the prevalence of the gene in the variants in the population. So it can't be a variant that's extremely rare, because then your patients would just be wasting their money. The variant would never come up. And the variants, and so that's where, when we're looking at variants, something that's considered common, just generally when we're looking at common single nucleotide polymorphisms, what's considered common is being more than 5% in the general population. And then lastly, all the genetic variants tested in our DNA Life test are what we consider functional variants. So they have a functional change on either the gene expression, so we can see from gene expression studies, because you carry that variant, there's either increased or decreased expression of the gene, or it leads to a change in how the enzyme might function. So an actual change that would occur in that overall biochemical pathway because of that variant. So that functional change is very important. And then obviously, and this goes without saying, we need to see the evidence. So if we don't have the evidence there, as exciting as the prospect might be to include one of these variants, we can't do it until the evidence is there, because how can we then justify what we are recommending? So that is just something very important to consider as well. So then we get to the report, and this is what it looks like. So I've just actually extracted one page of the first page of the genotype results. We've got a second page, so there are a lot of variants. Don't worry that you can't see it. It's just to give you an idea of what you might receive in the report, where you get your biological area being mentioned, the genes according to the biological area being listed, the variants related to that specific gene, and then the individual's genotype. And then, as I've spent quite a bit of time now, and hopefully your understanding of it is good now, on looking at those impact factors. And when you have the risk variant or the genotype, we assign that higher or that moderate impact factor. So then it should start to make sense that when you are trying to interpret the DNA health report, you want to look at where you see the ink, where you see those colors coming up in each biological area. If there's a lot of moderate and high impact factors in a specific area, that is where you want to focus, and then relate it back to the patient, obviously. And what we've done to make it a little bit easier for you is we have on our back end an algorithm for each of these biological areas. And based on the genotype your client has, they will get a specific score, and then you would come out as, you would see this in the report as showing a high, a moderate, or a low priority for that biological area. And that is based again on what we're seeing in the literature and the strength of the evidence behind those specific variants. So then what do we do with all this information? Now we can understand, okay, well, we see this report, there are a lot of genotypes, we see the impact factors, we see that for this person, they've got a high priority in lipid metabolism, methylation, inflammation, and oxidative stress. So now how do we kind of put this all together, and what does that really mean? So try to look at how, based on your knowledge of biological pathways and how they kind of interrelate, and if we're thinking about the interventions that we're going to provide, can we sometimes group them? And can we come up with 3 or 4 key biological areas to focus on in the beginning? So then this person has quite an unusual amount of high priority areas coming up. It doesn't always look like that, but you would want to be able to say, okay, I'm going to focus on the high priority areas for you, so you don't overwhelm your client and you don't get overwhelmed yourself. So identify the key 3 or 4 biological areas that you can see, either by looking at these moderate and high impact factors related to that specific part of the report, or using our cheat sheet where you are able to see the priority areas given to you. Based on then understanding which 3 key biological areas you want to focus on. So for this individual, it's going to be lipid metabolism, methylation, and actually inflammation and oxidative stress. If we consider the interventions around them, how they play into each other so clearly, we are going to just join that as one big pathway. So lipid, methylation, inflammation, and oxidative stress is your 3 key pathways that you could identify. Then based on those 3 pathways, what are the lifestyle and dietary interventions that you could put into place to kind of offset any weaknesses from a nutrigenetic standpoint? So giving an example here, if we were to take methylation, one of the priority dietary interventions would be to look at your folate-rich foods, so really upping the green leafy vegetables for that individual. So that would be a dietary intervention. A lifestyle intervention would be looking at really reducing stress, and another one would be reducing alcohol, because both of those are going to further deplete any B vitamins that are so important for methylation to work properly. And then so that would be your dietary and lifestyle intervention, and just an example of that. In terms of your nutraceutical recommendations, this is where if we are not able to meet requirements with diet and what we are taking in from food, we do need to look at what we can do from a nutraceutical standpoint. And this is where you would identify the key nutraceutical supplements that would be required to support that specific high priority pathway. An example with methylation, again, is a B complex in the methylated forms. The dosages would depend on who's sitting in front of you and what they're taking in from their diet and what other possible comorbidities they might have. And then the last of these 333 rules is to look at what phenotypic tests would you want to do in terms of biochemistry or functional tests that would help you measure a pre and a post intervention. So with methylation, would you want to look at homocysteine levels? Would you want to look at red blood cell folate and B12 levels to actually understand a little bit more clearly what is going on in that pathway right here and now? And so with this, hopefully this kind of step-by-step process, identifying the top 3 key biological areas. Based on those biological areas, you can put in diet and lifestyle interventions. A lot of that actually is given in the report, which you will see coming up, but also with our training, you get this information and you learn more about the nutrigenetic and nutrigenomic interactions that have been nicely studied around lifestyle, diet, nutraceutical, and then obviously there's always a measure involved, so you can get an idea of which phenotypic tests to consider. So then if we were to look at this report and just taking you through it practically, as another example, we've got all of your genotype table. And in this genotype table, we now want to see, well, where are the moderate and high priority areas? So for this individual, we can see there's a high impacted detoxification. We can see a lot going on in inflammation and oxidative stress with lots of color. We can see around your PUFA metabolism, salt sensitivity, and caffeine sensitivity coming up around, this is all related to your food responsiveness, and actually all plays a very important role in cardiovascular risk. So those very important and sensitive to cardiovascular function. And then looking at some of the others around food responsiveness is your gluten intolerance, and you might get some good indication of just doing some further tests around that. And then lastly, looking at your vitamin requirements, and we can see those vitamin requirements actually impact all of these pathways to varying levels. And we can see there's quite a lot going on there. So we would want to then, based on that information, we could identify that, that we see your detoxification, your inflammation and oxidative stress playing very important roles. I didn't put in methylation too much for this client, but we could look at it as a sort of a secondary one. But for this person, inflammation, oxidative stress, and the detoxification playing very important roles. And obviously seeing who's sitting in front of you. If this person was actually did a report like this, quite very similar to this actually, with a practitioner going through with them, and this was there was an individual with a lot of vascular issues as well as gut health issues, really complaining about gut health concerns, irregular and just very uncomfortable on a lot of medications to try and support it, low vitamin D levels. And so if we were to look at this kind of panel, we would definitely be able to pull this information across to the phenotype and try and relate it back to whoever's sitting in front of you. So identifying those top 3 biological areas, and then based on the biological area, if you open up the report, what comes up first is lipid metabolism, but you'll see each of the biological areas are actually listed according to the order that they're listed in this summary page. And there you see all of the information given to you. So you don't need to know all of this by heart. You don't need to remember variants and genes and what they mean and specifically what intervention is required just off by heart, it is given to you in the report, where we give a brief description of the biological area. We again give the genes, so it's right there in front of you. You don't have to page back and forth. And then your gene and genotype specific content is given as well, with the relative impact factors. So all of that is actually provided. It gives you an explanation of what ApoE is about. And because of that specific genotype, it again explains it and what you should do about it and what the literature is saying. And then what we do at the end of each of these sections is we highlight again the summary around that biological area. What priority level was it? Was it moderate, high, or low? The overall summary of the recommendations with any specific genotype content that's very relevant for that individual if they came up with that specific genotype. And then in terms of next steps, what biochemistry would you want to look at afterwards that would be relevant for measuring before and after intervention. So all of that is given to you. So you identify your 333, and then from there you can actually go straight into the report and get those recommendations right there in the report. And so to summarize that, you've got your 3 key biological areas. You identify your 3 key diet and lifestyle interventions based on these biochemical areas that you've identified. You identify your 3 key nutraceuticals and your 3 key phenotypic tests. Obviously, the 3 is a guide. It will sometimes be more, sometimes be less, depending on who's sitting in front of you and what they're willing to start off doing. And then just highlighting this image that nothing works in isolation. We always need to consider how one pathway might further drive the other. And something just to also highlight here is, you know, sometimes you might expect, when you look at a patient, we sometimes get an idea. We say, oh, this individual, they must be struggling with inflammation, the inflammatory genes. If we're going to get the report, I bet you all of the inflammatory markers are going to come up. And if you look, then nothing comes up in inflammation, but you see a lot coming up in oxidative stress and detoxification and your food responsiveness and your vitamin requirements. And remember, all of those pathways will impact inflammation, and especially if the diet and lifestyle isn't kind of bathing those genes in the right nutrients, the right environment to see good protection and good expression of your antioxidant genes, your detoxification genes, it's going to further impact on what you thought was the eventual problem. So again, just another really useful insight from DNA Health is where you are able to target interventions based on understanding where the variants are actually occurring. And so from identifying these diet and lifestyle recommendations, we give you further little take-home messages that you can hand out to your patient to make it easier for them just to remember which information is important for them and where they need to focus. So this is just after the results summary, where you get a report summary of what is important for them to focus on, whether it be the lipids, the detox inflammation, whatever it might be. We then give a short description of, again, what that area means, but kind of in very patient-friendly terms. So this is the consumer take-home message, where you can then highlight and scribble what you would want to do further. So we focus on the diet and lifestyle, and this is where you can further put in nutraceutical recommendations and functional and biochemistry tests that you would want to recommend for that individual. So that's really what the report entails, what you would get from it, and some of the useful parts of the report that you can hand out to your patient just like that, so that they don't feel completely overwhelmed when leaving the session. They've actually got something to read that's very patient, consumer friendly. To end off, I'm just going to go through a very quick case example and showing the utility for an individual or for you as a clinician in practice. And this is something that we often see coming in, especially if we're seeing executives, they've done their medicals at their relative companies, and then they come in And then they want to go a little bit further because something has been identified that they weren't too happy with. So this is a middle-aged male. He's married with 2 children. He's an executive in his own company. He's got high stress. He's a social drinker and occasional smoker. He has tendencies towards modern Western lifestyle, but he does also enjoy his fatty fish, his vegetables. So it's not all bad, but there are definitely things around refined carbohydrates and some of the kind of the wrong things coming in. But he's very open to tweaking. He is slightly overweight and presented with eczema when we saw him, which he states is exacerbated under stress and change of seasons. And he also has a bit of a sensitive stomach to any unusual foods, as he calls them. He has a history of cancer and heart disease in the family, and he came in because from his executive medical, there was they identified dyslipidemia, so he wanted to see if he could sort that out with lifestyle. When we did the report, 2 of the big areas that came out for him was detoxification and vitamin D metabolism. So those are just 2 areas in the report that really shone out. And I'd like to just go into what you would see if you were then to see, okay, well, detoxification as an issue. You would go into the report and look through what the results are showing, and we can see that in phase 2 detoxification, we see that is because of those high impacts, those variants, it's sluggish. And then we give all the recommendations there for you around what that means. It's a high priority. The recommendations around what to do in terms of diet and lifestyle and possible nutraceuticals, and then also next steps you might want to consider for this kind of person. So the SNPs tested in this panel are found on genes encoding your proteins and enzymes related to inefficient detoxification processes, or efficient if they're all working nicely, and thus can be a risk for, when we're looking at the research, they can be a risk for having higher DNA adduct levels. So that's pretty much broken DNA. We don't really like lots of broken DNA floating around in our bodies. It's been associated with cognitive decline. It's also been associated with certain cancers. So poor detoxification means DNA damage, which means cell damage, which means increased risk for cancer development. We see that it's been related to vascular issues, atherosclerosis, stroke, myocardial infarction. He has these family histories of cancer and cardiovascular disease. Also been related to atopic asthma and skin sensitivity. So again, looking at the individual, seeing what the priority is and relating it back to this person. So you can, by understanding that just looking at detoxification as a high priority, and if the environment isn't there to support this individual with this weakened pathway, we can see a lot of these issues maybe beginning to manifest. And as an example, there are certain mycotoxins that you can take from different foods. This is actually from quite an old study, but interestingly, another more recent study actually even done on peanut butter consumption and cancer, and the toxins that you get in peanuts, the molds that you find in peanuts. And it has been surprisingly high consumption related with certain developments of certain cancers. But it's a big but. It's related to whether you carry a GSTM1 deletion or not. So individuals who are exposed to certain toxins, mycotoxins, molds, other environmental oxidative stresses, they can manage them really well, and they go through phase 1 detoxification. They get then conjugated with your fantastic phase 2 GST family, and they have good antioxidant protection, and everything works out quite well. Whereas for an individual who is exposed to these toxins in high amounts, but they do not have the protective variants, the supportive variants to help with the detoxification, they may end up having a much higher risk for certain cancers. And in this case, it was stomach. In the other one, it was related to esophageal cancer. And so just what you would get, and I'm not going to go through this in a lot of detail, but what you would get from the pathway, from the training, is you would be able to get information on the background of how detoxification works and the nutrients involved to then support it both nutrigenomically and nutrigenetically. And so then from there, also what is given in the report, but what you can also learn from the training is what biochemical tests to consider. And that's why Nordic Laboratories it's so fantastic that you can just go there and it's a one-stop shop and you can get all of these nutrients, all of these tests to consider as well for an individual who is really struggling. And then from there, again, from the training and from what we've recommended in the report, you can go through this 333 around your diet and lifestyle, what nutraceuticals you might want to consider, and the follow-up tests you would want to look at. And so that would be the one area we would really want to focus for him on changing that modern Western diet, but focusing on those cruciferous vegetables. And then the other area that I wanted to just quickly highlight is on vitamin D metabolism, and showing how these high impact factors around the transport with GC and then the CYP2R1 of the activation to the active form of this nutrient can be so important when considering vitamin D levels and expectation on supplementation for an individual. We know when we're looking at vitamin D, it's as the sun is needed to activate it in the skin. We can get it from some dietary sources and obviously supplements. But why vitamin D is so important and why this actually I wanted to relate it back to this case is that it has been related to inflammation, to immunity issues, to vascular health, all of these things that again came up with this individual. So what we would want to see is based on these variants, what is the responsiveness to the supplementation that we're given, and will that improve some of the areas around immune regulation where he's obviously having an issue with that eczema around inflammation with the vascular system, the risk of cancer, also vitamin D being so important. So here, very simple in terms of the dietary recommendations and why, again, to just use this as an example, it should be so relatable to everyone, especially in the last 2 years, vitamin D coming up as such an important nutrient to consider around inflammation and immunity. So looking at your increased intake of vitamin D-rich foods, your sunlight exposure, what you're going to do from a nutraceutical, and then doing the follow-up tests. And again, with these genotype results, what they've been related to is a change in the incremental increase in with supplementation, looking at your vitamin D levels. So you might expect to see a slower incremental increase for an individual who has these variants. So just giving that really nice insight based on what you're seeing in the biochemistry, but then what you would be able to see in the DNA health report. And so for that individual, detoxification and the vitamin D came up as one of the big issues that we wanted to focus in on, and then obviously related back to that patient. So always keeping in mind that all of the tests, all of the functional tests, all of the genetic tests need to be considered according to who is sitting in front of you within the context of the patient, using all of your other experience as well, and not when we're looking at genetics, just forget that we have all of these other expertise and this other knowledge that we can just use genetics as a tool in our toolbox, and sometimes not completely kind of get frazzled with understanding the kind of the science behind the genetics, and rather just using the reports to put it into practice, and it's all there for you. And obviously using it as now just emphasizing in the context of the patient. So that is me on just an introduction to DNA health. Why, when, what, and how.
Excellent, Helen. Thank you very much. Very impressive, quick introduction. Managed to go through the literature in a short time, but very convincing, I think so. And again, for those of you who have not done the education, I strongly encourage you to do the education. You do learn a lot, and also consider looking into the test pairing, like that you get certain information from the health, and that leads into some something else that you want to know in the body, so that you have the genetic information, but then you have the phenotype information as well, and you can match it and explain why things are the way they are. So it becomes very specific what you do when you work with the patient. So not that many questions has come up. So I think people have been very impressed with your introduction. And I think I'm just gonna open up for an invitation for your webinar at the end of the month, where you're gonna talk about DNA risk, which is a bit of a different genetic test, because it's looking more into autoimmune conditions. So I'm looking forward to getting some information on that test for sure. Yeah, so I think that's what I'm going to say now. I will send out the slides. Someone praised your slides when you were talking, and I think they're very excellent as well. So, but they will be shared with you. And the recording will be shared as well. So thank you, Helen, very much, and see you soon.
Thank you. I'll see you
Bye,
very soon.
everyone.
Thanks so much for having me.
Welcome. Thank you for joining us.
Bye.
And thank you, South Africa, for the electricity to be on so that you could join us.
It's not.
Okay.
It's going to die.
Oh, you're on battery? Well done.
Yeah.
Well done.
Just made it.
Thank you. Bye, everyone.
Bye.
Bye.
If you do not already have an account with us, please contact our team and we will assist you.